We do powder form, we do liquid form. So we can mix them in the feed. We can also add them to water. So it's totally depend on what the industry looking for. So if the commercial setting, they have like a automatic feeding system, or they can like mix to the feed is much easier for them. So they can mix like the powder form in the feed.
Mhm. Welcome to the Feed Science Podcast. On behalf of Wisenetics, my name is Adam Fairnoltz. I'm coming to you from North Carolina State University's Animal Food Program. Glad to have you guys listening with us today. My guest today is Dr. Yanhong Liu. She is an associate professor in the Department of Animal Science at the University of California at Davis. Dr. Liu, how are you today?
I'm doing well. So how are you, Adam?
I'm doing just great. I'm really glad to have you join us. If you wouldn't mind, introduce yourself to our audience and give us just some of your background as to how you became a professor there at Davis.
Hi, everyone. This is Yanhong. I originally from China, so I got my bachelor and master degree from China Agricultural University. So then I moved to US like back to 2008. It's a long time ago. So I did my PhD with Dr. James Pettigrew at University of Illinois Champaign-Urbana. After that, I also did another 3 years postdoc with Dr. Hanstein. So this is where I become a true swine nutritionist. So
Mhm.
after that, I moved to UC Davis. It's actually in 2016 January. So I moved to Davis as assistant professor, mainly focused on swine nutrition. But since I moved to Davis, my research kind of like abroad to different area. Swine nutrition is still my major research focus, but I'm also trying to expand to poultry nutrition, and also we are doing lots of research with antimicrobial resistant development.
Okay, very good. So primarily looking at swine on the nutrition side, and then, like you said, branching out a little bit there into the poultry spaces, some of the antimicrobial resistance. I know that a fair amount of the work, and based on some of our prep as we lead up to the podcasts, you had mentioned that some of your work has had to do also with a lot of like weanling pig type of work as well, things with organic acid. So if you wouldn't mind, give the audience a little bit of a of some specifics of some of the work that you've done there at Davis so far.
So the E.coli research actually started with my PhD training. So I did my first E.coli challenge with Dr. Pettigrew. So that's where we start the research idea, the E.coli challenge model. So since I moved to UC Davis, California is not a really big swine state, so we don't have a lot of a commercial farm around, but we have our really nice swine research facility. So we can get pigs here, but we cannot do like a really large performance trial. So that's why we're back to our regional focus to the post-weaning pig and newly weaned pig and E. coli diarrhea. So this is where I start my research with E. coli. And how far, I think so far we are doing great. So uh, we have very consistent model with E. coli challenge. So we have many different strains from California and also from Canada and from Midwest. So we have a different E. coli strains covered in different area. We use this E. coli challenge model and try to figure out if we can promote their gut health and if they can bring beneficial effects on performance and disease resistance. And as I mentioned, like we focus more on post-weaning pigs and focus on more modes of action. We explore different type of feed additives, including organic acids, like you mentioned. We also do other lots of other types of research projects covering like probiotics, prebiotics, functional amino acids. And currently also we are working with some new like very novel ingredients or crafted oligosaccharides.
Sure.
But back to the organic acid, maybe we can talk a little bit more. So
Hmm.
I'm very excited with this type of feed additive. We are doing lots of research. We have a collaboration with Eastman. We have a collaboration with other industry, and we also get some USDA funding support and also some nonprofit funding support. We can discuss a little bit more later.
Eastman serves veterinarians and nutritionists in agrochemical and animal health industries by helping them select, evaluate, and implement innovative nutritional programs. Eastman works with your team to customize your gut health approach in feed and water. Eastman's approach addresses nutritional and bacterial challenges and finds new ingredient preservation and hygiene solutions. Explore ways to accelerate and innovate your programs. Contact the Animal Nutrition Team at eastman.com.
Being the Feed Science podcast, we'll try to stick kind of close to those things that you mentioned on the different things that you're feeding, looking at trying to control for disease and deal with the E. coli challenge based on the model there that you've created. So when you say organic acids that you're feeding to the animals, a couple questions that first come to mind as kind of the feed milling guy here are, what organic acids are we talking about? And are we talking about dry forms, liquid forms, both? And what have been some of the advantages, disadvantages, challenges of using them in the manufacture and the feeding of those products to animals so far?
For the forms, so we have many different types, and also even for the products, we have many different choices as well. So we in our research we use a dry form, powder form, and we also have a liquid form. So it's because, like I mentioned, we are doing research in the research unit, so very small scale.
Mhm.
So any potential benefits for the commercial production system, we have to do large scale trial.
Mhm.
But in our a small scale animal trial, we do powder form, we do liquid form, so we can mix them in the feed. We can also add them to water. So it's totally depend on what the industry looking for. So if the commercial setting they have like a automatic feeding system, or they can like mix to the feed is much easier for them. So they can do they can mix like the powder form in the feed.
Mhm.
If they have like a water, it's very easy to supplement the feed additive into the water system. They can also do that. So we cover both. For the different types of organic acid, we are doing some research right now with sodium butyrate. It's kind of like a chemical form. So we also have other types of organic acid, salt form. We also do monoglyceride, like a glycerol form of short chain fatty acid and medium chain fatty acid. We're also doing some research with a blend or mixture. So all different type of forms. Compare with organic acid, like butyric acid, valeric acid. So they don't have a very pleasant
Mhm.
smell. So that's why the salt form and the glycerol form, so they have a more advantage for like they don't have that bad smell, so they can easy to be mixed into the feed. So they will not reduce the feed intake. So they are very stable. So they will not like um, volatile, or when we mix to the feed they will disappear very soon. So stable, heat stable, and they can stay store for a long time, and they are also like a no unpleasant flavor. So that's a major advantage. So that's why we are focusing on the glyceride form of organic acid. But like the salt form already used in the market for many years. So salt form is kind of like our reference.
Mhm.
So we can compare with a salt form. We can also compare with other types of organic acid we use as a feed preservative. So, but our final goal or research question is if we can use glycerol form to replace salt form, so they can have a better efficacy. So they can probably has a better impact on the overall gut health.
Sure. So it sounds like some of the advantages that you see as far as the handling, let's say, of the material, it ends up kind of being beneficial both for the manufacturer of the feed, in that it's something that's maybe not so difficult to deal with, doesn't have some of those side effects, maybe not the right term, but the issues with dealing with them, with the smell or the makes your eyes water. You don't really want to inhale it, all that kind of stuff. But you also see the same thing then on the pig side. They know when it's there in that form and may or may not want to consume it, but in the form that's a little bit more pleasant to deal with on the manufacturing of the feed side, it also is turning out to be something that the piglets actually are being a little bit more receptive to. Is that correct?
Yes, that's correct. So, for the short form, so if we think about the GI tract of the weaned pigs, so short form, so whenever they enter the stomach, they may dissolve, so they may disappear in the stomach. So
Mhm.
when we think about post weaning pigs, and especially the specific disease, E. coli induced diarrhea, so the disease mainly target the lower small intestine, so lower segment of the small intestine. So the glyceride form, so we are looking for more products can be delivered into the lower part of the small intestine. So they may cover the whole GI tract. So they may, have a better results compared with a short form. So it's kind of like we can say they are slow release form compared with
Mhm.
a short form. So that's a major goal. But they also have other different modes of actions. For glycerol, or glyceride form, so they are more like a lipids, so they can escape from the stomach digestion, so they can enter the small intestine.
Okay.
So that's another benefits compared with the short form.
Okay. Yeah, kind of the same idea as like our rumen protected amino acids and things like that. You've got something that's getting further down the GI tract. Your the feeds that you're feeding that you're adding these acids to. Are we talking meal feeds? Are we talking pelleted feeds? Some of both. What's the feed form that you usually feed to the pigs?
At the current stage, we only work with a mesh form, so
Okay.
it's not pellet. So we didn't test if we mix the products and then pelleting. So how much the products are still available. So that's I mean, maybe the future research, but currently we have our own like a small mixture. So for doing research, we can do everything by manually, so we can, it's very easy, we mix our products and then into the mesh feed.
Excellent. You said in addition to doing then like the organic acids, you guys have also branched these experiments out into some other types of feed additives. You mentioned probiotics and some other novel things. So, what are some of the interesting things that you've worked with, and that what kind of results are you seeing from those? On their own, but also in comparison to the organic acids, which has maybe been something that we've thought about as a solution for a longer period of time.
Just to clear the question, so we don't compare the products versus products, because
Okay.
so far we never done any research to include different types of the products or feed additive.
Okay.
So we never done organic acids versus probiotics, because uh, our hypothesis, like they have a different modes of action,
Mm-hmm.
so they
Sure.
can probably bring different benefits to the GI tract. So but we did some research with probiotics, bacillus. So bacillus strains, so they are very stable. So they are very easy for the feed mill to manage. So they can either mix into the feed or with the powder form, or they can after pelleting they still very stable, has a high efficacy. So we did several research project with a bacillus subtilis. So the results are very promising. So we use carbodox as positive control. So their efficacy is very comparable to carbodox. So that's
Okay.
one thing we are very excited. So currently we are doing more research and try to explore if the pigs they receive Bacillus subtilis versus antibiotics. So if their gut microbiome diversity changed or not, if their AMR gene development changed or not. So more research currently focus on, so if the feed additive, so they have a very comparable performance or disease resistance benefits compared with antibiotics. So if they can also slow down or reduce the antimicrobial resistant gene development. So that's our current research focus in one project. Another one, and we are very excited because we have our lab is currently involved in one really big project. Like I mentioned, it's funded by a nonprofit foundation from Denmark. It's called Novo Nordic Foundation.
Mhm.
So this project is mainly cover post-weaning diarrhea.
Okay.
So it's completely matched with our research focus. So focus on post-weaning diarrhea, but we have a more than 30 faculty or research scientists from Denmark, from the Netherlands, and also from UC Davis. So we have a really big group focused on the post-weaning diarrhea, post-weaning pigs. So we have a group of faculty focused on the host, the pig, and understand their biology, understand how their GI tract development during this physiological stage. And we have one group of faculty only focus on the microbiome. So understand the microbiome change before and after weaning. So how the microbiota play the important role for the overall gut health of the post-weaning pigs. So our groups at UC Davis, we focus on nutrition. So we work with different type of nutrition strategies or nutrition supplements. We try to understand if any nutrient or nutrient change or any supplements, so they can modify the gut microbiome or mucosal immunity, and then eventually they bring the benefits to the host.
Okay.
So that's another one we are currently working on. So it's also related to organic acids. We have one small project with organic acid, actually funded by this big project.
So yeah, a lot of different things kind of all focused there on the kind of the lower GI tract and diarrhea and things and the pigs and the E. coli, but coming at it from a number of different directions. That's certainly very interesting. Once again, my guest today is Dr. Yanhong Liu. She is an associate professor in animal science at the University of California at Davis. So you mentioned a little bit that you are looking at the potential for products to do the same thing as what we've seen antibiotics do without contributing to any of the antibiotic resistance that we might be concerned about. For those that everybody's heard antibiotic resistance, right? Everybody has heard about why this is a problem or not a problem, and depending on where you get your information from, it may be good information or bad information. So from your perspective, based on the work that you've done, but also as hopefully everybody that's in the audience is aware, when we do these kinds of research works in academia, there's a lot of literature review that goes into deciding how to set up the experiments and then also how to compare their results to what's been done in the past. Can you give us a bit of a explanation as to why it's so important for us to find things that we can use similar, from the feed milling's perspective, it needs to be something that we can use easily, like an antibiotic that comes in a premix that we can put into a bin and easily put into feed. Why it's important that we have those kinds of products that could easily replace something else in manufacturing, but not lead to the resistance concern.
Just one more concept clarification. So
Sure.
we're not doing research to replace antibiotics,
Mm-hmm.
because we know like especially in livestock industry, antibiotics still very important to control and to especially to treat disease. So our research, I think many labs, their research is not to completely replace antibiotics. So when the animal when the animals are sick, we still need to use, especially when they have a bacterial infection. So we still need to use antibiotic as a treatment. So our goal is trying to, uh, make the pig stronger. Raise the pig stronger. So when they are stronger, so they have a stronger disease resistance. So then they still get some pathogen exposure. So they may not get like a very bad infection. So they may need not receive like antibiotic treatment.
Mm-hmm.
So that's a goal. So that's another reason we focus on post-weaning pigs, because it's very similar as a weaning calves. So like maybe your research area. So For this specific stage, piglets, they are very easy to get enteric infection, so they need the antibiotic treatment.
Mm-hmm.
So they are the special stage, need lots of antibiotics compared with other physiological stage. So that's why post-weaning pigs or manage post-weaning pigs health is very important. So if we back to AMR, so antimicrobial resistance, so because we use antibiotics in the feed as a growth promoting purpose for many, many years,
Mm-hmm.
so the AMR development is a truth. It's not like so it's already present in the society. It's already affect the human medication and human or public health. So that's why we ban the antibiotics in feed as a growth promoting purpose. So we cannot mix the antibiotic in feed anymore. Just try to promote the animal growth or prevent disease. But we can still use lots of antibiotics to treat sick animals. So that's one thing. Like we have to, our audience has to be very clear. We can still use antibiotics, but we are trying to use less,
Mm-hmm.
so not like as much as we do in the past. Um, for the AMR, we don't have a tons of literature review in livestock industry yet because AMR is actually uh, it's not new focus, but there were not lots of research focused on AMR in livestock industry because the restriction just published in 2017. So we have now we have like 6 or 7 years after the restriction or after the regulation. There's still lots of research ongoing and try to figure out how bad the AMR actually spread from the livestock industry. But there's no like a very clear. We have a clear evidence we contribute livestock inter contribute the AMR development, but how much compared with the human side, we
Mm-hmm.
don't know. But the one thing like we have some evidence or some like data. So if any of you listen to this podcast, you can go to NAMS. So we have a special NAMS monitoring system monitored by the USDA. So if you go to NAMS website or database, so you can see how the AMR present in our food in the grocery store. So currently we are involved in one project, a very exciting project. So we're helping NAMS or FDA, the FDA related to NAMS. So we're helping FDA to collect meat samples or seafood samples from grocery stores in California and also in Hawaii. So then we send the sample, we isolate the bacteria in this meat sample or seafood sample, and then we send the bacteria isolate to FDA. They do the whole genome sequencing, and then they figure out the antimicrobial resistant gene presence or not presence. So the results are pretty bad. There are lots of AMR gene present in our regular food.
Mm-hmm.
So it's not like a very small percentage. So we test the E. coli, we test the salmonella and enterococcus and campylobacter. So very common bacteria, they may be commensal bacteria or pathogen, but they have our meat samples we collect from grocery store carry substantial amount or percentage of this AMR genes.
Good reason to thoroughly cook your food if there wasn't a enough a reason already, right?
Yes, that's correct. So the raw because we deal with the raw samples or raw
Mm-hmm.
meat. So when you buy buy the meat from grocery store, any grocery store, so you have to fully cook before consume them.
Yeah, we don't want to get we don't want to not only do we not want to get sick from E. coli, salmonella, on and on the food pathogens that we're concerned about. We are concerned potentially of whether or not those particular bacteria could potentially carry a gene that would make it difficult to treat that infection too.
Yes.
Again,
Mhm.
cook your chicken and cook your pork like you're supposed to, and some of that problem will go away. So it's another good reason to do that. I think it's also, and I'm really glad you clarified the point of we probably do focus too much on this idea of maybe trying to get we don't want to use antibiotics at all anymore, and I'm glad you clarified that, because that's not really the case. What we want is for the antibiotics to work when we do need to use
That's
them.
correct.
Therefore, we are trying to come up with other solutions to keep animals healthy or to treat low levels of disease if we can, in ways that don't involve the antibiotics, so that when we have an actual disease outbreak that can be treated, the tools still work. I think is a fair way to say that.
Yes, we are trying to protect more useful tool in our toolbox. So
Right.
antibiotics is one very important tool in the livestock production system.
Yeah, I think it's a great way to look at that. So as we kind of come to our end here of our 30 minutes, we've talked about a lot of interesting things, and I appreciate all the information and talking about the different things that we are looking at when we are trying to deal with, especially in that post-weaning pig, but potentially other species and other ages of the pig and everything else. These ideas of tools that we may have to address gut health, and we've got organic acids, and we've got probiotics, and we've got antibiotics, and they all serve a different purpose, and sometimes they work together, and sometimes it's this one, and then if that doesn't work, then this one can work. From the feed milling perspective, we may have multiples of these products in our facilities in order to use them depending on what the need of the animal is. So one of the questions I'd like to ask as a follow-up here as we end is, if people want to get more information on this and they want to get it from a reputable source, they want to learn about what probiotics are, or the organic acids are, or things related to antimicrobial resistance, what would be some you mentioned one already there with NOMS, but what would be do you have in mind any other resources that you would say, hey, if you want to learn more about this, here's a good reputable place to go learn about these types of topics?
So far, that's that's something we have to improve from academia side. So, like, we are doing research, and our research, I think we are doing important research for the swine industry, but we somehow lose the connection with the public.
Right.
So that's one way we have to improve in the future, like how to, you know, let the public understand our research and quickly get what we are doing, and also understand our research finding. I don't have a really good resources to point out at current stage. So I know the new NRC. I'm also involved in the new NRC swine. So
Yep.
we are trying to provide some information for feed additive or feed technology. So that's probably somewhere they can, when we publish the new revision of NRC. So, but like I mentioned, it's more for grad students, more for industry people, but
Right.
for public, so they may not know NRC, so they may not find the information from NRC. I know lots of universities like Kansas State University, so they have a really good extension group. So they always post some like a very general information for feed additive or basic nutrients for swine. So that's
Mhm.
some place audience can go and can find the general information. And Iowa State, so they have a really good extension center, swine center as well. So, but that's the information like when you ask like the reputation, or they both of them they have a really good reputation. But I mean,
Mhm.
for the public side, so I think we need a more podcast like this type of the form. So
Right.
so then the public audience they can quickly learn the problem in the different like livestock industry and what we are doing right now in the university. So then we have to build a connection, so then they can get more recommendations or guidance or understanding on the agriculture.
Yep, that's right. It's unfortunate, but it's very easy for someone to take a truth like you shared before of, okay, we're doing this research, and yes, we found resistant, potentially resistant bacteria in a sample of meat, right? And when that's kind of the end of the information, and or that's all that somebody from the public hears, it then sometimes balloons to something that becomes not a necessarily a truth, right? Where it's, well, and that's there because of this, and there because we're feeding all these wrong things and the animals, and then people start making things up, instead of actually understanding the true reason why those things are there, understanding things very simply, like, well, even if it was resistant, if you killed it when you cooked it, it wouldn't matter because then the resistance wouldn't spread. So make sure you cook your food, and this doesn't necessarily have to become a problem anyway. And figuring out ways to share that information with the public is very difficult. Like you said, we've got a lot of good extension, a lot of universities out there putting out very good extension related things. They aren't usually the first thing that comes up when you do a Google search for, antimicrobial resistance in my food. You get some other website that you may not be good. So no, I think you're absolutely right. I think developing our resources there is something that we all need to focus on a bit.
It's time for our famous three.
Last couple questions, then we're gonna flip this a little bit away from the science and just ask you a couple questions. So, as you have grown in your career in academia and have moved up through graduate student to postdoc to assistant professor, now an associate professor, and someday on the way to being a full professor, are there any particular resources that you have found particularly helpful in your career, not necessarily even on the science side, but as far as, personal and professional growth that you would like to share with the audience of, organization you belong to or books that you've read that you found really helpful?
I think to me, I have to point the university support. So I
Hmm.
since I moved to UC, not UC, like US.
Hmm.
So I had a really really good mentor and advisor. So I think that's a very important for any grad students or new career researcher. So a good mentor is very important because that's something like can motivate you to go further. So and can make you love like doing research.
Yeah.
So that's one way. I think that's why I start. I want to stay in academia. So after I moved to UC Davis, I think I get really really good support from the university, from the department. So UC Davis, like I mentioned, is not a really strong swan state.
Hmm.
So, but we have a really good collaborators. So we are the biggest per consumer. So
Right.
we know the consumers need, and we know the market, and we know like what the public concerns are. So that's a reason like we have a really good collaborations and inside UC Davis and also outside. So we can very easy to find collaborator, find anyone to support you if you have a good idea, if you have a really novel idea to support the industry. We also get a really good support from National Pork Board.
Yeah.
So I got some small project funding support from Pork Board. So that's why we realize our research is important for pork producer. So we are not just doing research. We are not just doing like a basic, uh, you know, research and then our results nobody care and no use.
Right.
So the pork board support is very important. It's kind of like a very big encouragement for us.
Hmm.
So our research is needed by the pork producer. I think that's very important. So for anyone work in your own society, so if you can get some funding support from your commodity, so you're like a stakeholder or the one like the farmers, they really need this type of research. That's big encouragement for any early career scientists.
Yep.
So we also get a really good support from the industry. So that's another thing like we think our research is useful. So we're doing research, we do proof concept test, so then they can find some large scale trial or test, and then finally the products can be used by the industry or by the feed industry. So that's another thing I think really really important. So um, I think that's all the resources we get a lot. I get a
Yeah,
lot from everywhere. So, family support, friends support, and also the most important, the university really support any type of research. So no matter it's from industry or nonprofit foundation or the government. So we are not only focused on the government or federal funding. So anything useful and also match with the agriculture, our agriculture mission. So we will go for it.
those are two really good points. I think the point of and it whether you be doing research in academia or research somewhere else, or even if you're not doing research, making sure that when you go into a role professionally, that you will have the support to do what you need, the that you'll be given the financial support, the time support, whatever it may be to be successful is something that everyone should be looking for when they're going into a career. And also, I think your comment about mentors is really well taken as well, and it comes from both sides. It's when you are growing in your career, you should be looking for mentors, people that you respect. You like something about the way that they are handling their business, their career, so they're your mentor. And then those of us that are growing in our careers also need to be prepared to mentor those below us when they ask for it, right?
Yes.
It's easy to go and ask for someone to be a mentor. Sometimes it's harder when someone comes to you and wants you to be their mentor, and you think, wait, why would you want me to be a mentor? And sometimes that's a hard shift to make. But if we want to have good ones for ourselves, we also need to be prepared to be a mentor for someone else. So I think that's a really excellent point.
That's actually reminded me. I have to add one more
Okay.
for the students. So
Yep.
we have a really, really good students, grad students, undergrad. UC Davis has the biggest largest animal science department. So we are the largest one in the US. So we
Hmm.
get a lot of good undergrad as well. So undergrad support and the grad student support, so they are the most important component in our research program. So
Sure.
we got a really good support from the student support from the department and college as well. So that's why even California is more expensive compared with other states. So we can still support several grad students in the lab and still support their research ideas.
Absolutely. All right. Well, again, my guest today has been Dr. Yanhong Liu. She is an associate professor in the Department of Animal Science at UC Davis. Dr. Liu, thanks very much for your time today.
Thank you, Adam. Thanks for the nice conversation.
Yeah, it was my pleasure. Once again, on behalf of Wisenetics, my name is Adam Farnels, coming to you from the North Carolina State University Prestige Department of Poultry Science in the Animal Food Program here. Thanks for listening, and we'll talk to you the next time.